All right. So now we are getting into some of the whole environmental and sort of experiential space that I talked about before in terms of the influence of experience on genes. And the study of how experiences and behaviors influence the expression of genes is called epigenetics. And this is just a huge, sort of hugely emergent field. It looks at molecules and systems that control gene expression without altering the DNA itself. And so this is really where a lot of the action is at. Aspects of our DNA have very small, like there's not too many sort of our DNA is largely similar. It's the expression of our DNA that's going to be really, really quite different. And so epigenetic systems, they actually help control how cells divide, differentiate during development. And aspects of epigenetics is also how stem cells develop into all the types of cells in our body, even though the DNA is the same. And so that idea that, all of our cells are going to come from one cell, together, that are created, by combining sort of the DNA from both biological parents. And and so this, bringing these, you have all the same pieces, but then what happens? How does this epigenetic system will then help the cell sort of develop into, okay, you're going to go become a pancreatic cell versus a skin cell, and the DNA is the same. So it's just the expression of the DNA at particular key times to create proteins and this and that would actually build us, and thinking that we all come from this single cell. So what are some of the things that are acting in these epigenetic systems? So when there are methyl groups, these are molecules that tag DNA transcription to kind of begin and to stop. And they can cause the DNA to also be wound too tightly, preventing transcription. So you can see that these are, like basically regulatory. Then you also have the acetyl group, and these can loosen DNA, allowing for transcription. So you have to have both of them. And here's a little bit of the space of how they work. Okay, so this just a little bit. All right. So here's some of the basics of the biology. So here we have our chromosome, and these are affected by various factors and processes. And so this is and then these are known as these epigenetic mechanisms. So these can include development, which would be in utero or childhood, things happening at particular times in utero, which would be sort of critical phases of development, but then also in childhood, there are particular critical phases. And if, things not happening at some of those critical phases or happening differently can have really significant downstream effects. So, well, when you're going to, replicate this DNA or access it for transcriptions or information on making proteins, then it unwinds. And then it unwinds, here, and you can, but slowly it'll be unwound. And then it will open. So some of these would be, again, this particular strand. So here, this is a methyl group that is going to, is attaching to the helix right here. And so DNA methylation, this is the methyl group, and it can tag the DNA and activate or repress genes. So it can, if it's going to tag it and opens it, then that would be something to go ahead and include sort of replication. And where if it's going to repress it, then it would probably try to stop it from uncoiling tighter, so that it would be less likely to activate. So the histones are the proteins around which DNA can wind for compaction and gene regulation. So here's these things right here. These are the histones, and I guess this is a histone tail. Here's part of the gene, okay, the particular interest in the gene. And it's usually, again, this is going to compose these base pairs that we talked about. And then DNA that is accessible or inactive is sort of further, it's harder to see, is basically about other parts of this that are not getting activated. So you can just see it pointing here. It's a little harder for me to see. You probably see it better on the textbook. So these basically are in this position to then they can start to unwind or not. And so here, when they start to unwind, then this piece of the DNA here becomes accessible. And okay, so this is, now I get what I'm saying. So right now, the DNA wrapped around here, this is what they're targeting. Right now, the DNA isn't accessible. And now when it's open here, it's accessible. So so then this is at this point when it unwinds, then you can activate the gene and you can engage in transcription and creating a protein. And so and then this here, which you can have on the end of these histone tails, could be an epigenetic factor. So these could be cancer, autoimmune, sort of mental disorders, diabetes. These can be some of the outcomes of these epigenetic factors. And they can attach to these and affect then how the histones in the DNA unwind. So as you can see here, the histone modification, the binding of epigenetic factors to histone tails alters the extent to which DNA is wrapped around histones and the availability of the genes in the DNA to be activated. So these epigenetic factors, they'll come in and say, all right, we're going to influence this and say that this piece is going to be really unwound, like really wound tightly. So even though you have it, you're just really not able to access it a whole lot. Whereas these other ones going to become, are going to activate. So things that can cause really different significant shifts are like experiences of significant trauma. So the impact of trauma at any point, but particularly the younger you are, is going to have more downstream effects. And traumatic experiences can really alter aspects of gene expression and really, but also depending upon how they're processed. So this is the type of thing also where experience come into play. Like two people can go through the same traumatic event, but not have the same outcomes. And it could have been from things before, could have been from like their experiences before, could be from genes, it could be from elements of behavior in genes, could be the support that they get afterwards, so there's all sorts of things, just giving you an example. All right. So a couple of other sort of examples of studies and research in terms of epigenetics is licking mouse pups. And so this is the idea that attentive mothers can actually affect methyl groups near the GR gene, which is in these mouse pups is related to the activity of glucocorticoids, which is cortisol, and these are stress hormones. And so mothers who are attentive, and actually sort of affect, like grooming their babies a lot, and licking them and giving comforting things, they the mice with attentive mothers actually handle stress better as adult mice. And so, and then, well, where does this attentive mother element come from? From this, from the parental rat. So one, that could come from their own experiences of being raised with an attentive mother. And that attentive mother can be that way in terms of genetically, or just learned through experience, which is actually a lot of the case with parenting. So those types of things can actually really affect, in this case, the experience of the mother sort of mouse pup relationship. that affects stress in later in life. And we see the same thing for relationships in humans. Now, there's also the town of Over Overkalix, yeah, hard to say. They kept excellent records in this very small town, and it's fairly isolated. And because of that, they kept these, like extremely detailed records of health and birth and death and all that kind of stuff, and including elements of the years where there was feast and famine, because it was so isolated that they really couldn't bring, things really couldn't be brought into, to the area. That it was like you are prepared, or for some reason, could be disaster, whatever, you're not prepared. Could be, just, poor, poor crops that year, or poor, things in the area, because this was sort of this was a very isolated group. And so, in looking at these over time, all of these records and the things that have happened, they found that the descendants of children that had experienced famine had less heart disease and diabetes. And so this may seem a little bit counterintuitive, but thinking about how heart disease and diabetes and all things are really related to diet, and in the sense of, and what could have happened during a year of famine, there's all sorts of things that could, an individual could learn from that, how to think about food differently. So it's like, you don't really know if this is genes or these environmental influences that are coming in and causing particular experiences that are then having these downstream effects. So in epigenetics, it's actually really hard to pull apart the nature versus the nurture, because it's pretty much always both. Now, in terms of the rise of sort of understanding these things, has also brought a rise in genetic testing. So there's multiple types, and I'll sort of mention some of them briefly here. Again, these are in the textbook. So there's cell-free fetal DNA testing, and this is basically for test of the mother in early pregnancy, and it's looking, for potential genetic disorders in the fetus that could be detected through the blood. And so that's this element of the cell-free that you don't have to have anything particularly invasive. And then you can also do carrier testing. This is a test prospective parents to see if they are carriers of recessive alleles for disorders. There's some pretty common ones that are really important to sort of look at in terms of even just basic metabolic processing and development, and so they can do those types of things, and these are really typically for stuff that is quite debilitating. Then you can also have pre-implantation genetic diagnosis, and this is where you want to try to find out for couples like ahead of time of what might, of like, let's test sort of, if we're going to go through and have our fertilize our eggs and try to, make a baby and go through the in vitro fertilization process. Oftentimes there are elements of that are again affecting the reproductive process, and to test some of those things, they'll do carrier testing, they'll do blood testing, and then depending upon sort of the genetic history, they can do this pre-implantation, this PGD, where they will try to they will fertilize several eggs in the lab, allow to develop to the 8-cell stage, test one cell, and implant the ones without the disease or those that are not carriers. And so this, then can be sort of a borderline for, again, trying to prevent diseases that are really metabolic, like really, debilitory in terms of like physical pain, blood work, body, the ways that food is processed, so these are things that are often left up to the to the doctors and the parents to decide, and based upon their own resources. But then it can also, what happens if, then you're trying to prevent, certain types of genetic outcomes that are maybe not quite so bad, right? So another type of genetics research that we have going on that, again, is really quite popular, because everything with genetics is popular a bit right now. So you have genetic engineering, and this is changing the DNA of an organism, creating a genetically modified organism, so a GMO. And so this is different from some of the other ones that are really looking at, just the expression. This is really physically changing the DNA, so changing the recipe. And most of the plant food we eat actually comes from GMOs, and most people don't know this, but it is but having these things genetically modified, it really affects the breeding, like long term, and they're bred for, like not necessarily taste as much, but for how they would look, in terms of beautification and selling in the store. So so how genetic engineering, you don't know actually like what the goal of is of it, and those things that are really important to sort of look into. Another form of genetic engineering that is relatively new, but with incredible potential, is CRISPR. And this is a new way to accurately and easily change or alter DNA. It's quite a complex machine, and there's all sorts of information on it to get to know more. I actually read a fabulous book on CRISPR and its efforts to try to revitalize the woolly mammoth. And they went through basically the development of CRISPR and using like really factual information, but then kind of taking it into, fiction in terms of the types of conversations that occurred, and developing some of the tech that was involved. But really, really great book if you want to read something really entertaining and knowledge building. It's called Woolly. And then you have transgenic organisms, where these have genes inserted, changed, or removed. And what is most commonly sort of referred to, these are sort of as the knockout mice. And these are rice or animal, like knockout mice or rats, are animal models for which research with targeted gene remove or sort of labeling or changing. So to test, again, specific roles of specific genes in this process. All right. Now, behavioral genetics is actually like a huge field, and probably, epigenetics is, to a certain extent, falls within the space of behavioral genetics. And then genetic engineering, some of it does. Behavioral genetics is like a sort of all-encompassing field within the genetics sort of psychology space that is specifically investigating the genetic influences of behavior. It often uses twin studies, so the monozygotic twins, which are identical, and the dizotic twins, which are fraternal. And so they share 50% of genes on average. So just like, two regular siblings. So then you can look at the differences in inheritability between them, between the twins, to identify how much certain conditions are heritable. And so heritability is something to kind of be really important to understand, because there is some misinformation about it. So heritability is a statistic. It is a statistic looking at the proportion of variation that is due to genetics or the environment. So all of our particular features, we have, there's, a range of heritability. And so what is the likelihood or proportion of variation, that is due to a gene or the environment, so the more something is due to the environment, sort of the less heritable it is. And the more something is due to a gene, the more heritable it is. But it's really also can be, it's just a statistic in terms of, okay, here's the proportion of variance, 50% or something in only this particular gene that, and so heritability is also oftentimes really difficult to assess. And using twin studies to look at correlations on physical and mental and sort of behavioral measures. And I think actually this is more sort of, here, there's the concordances. And then concordances is looking at probabilities that related people will develop sort of neuropsychiatric disorders. So aspects of heritability in here as well. So the spaces where heritability is dangerous is going back to some of the earlier parts in the chapter where people are attributing, again, more things to genes and more behavior to genes than is generally warranted. So these are sort of like the ultimate and the genetic determinism. So like high inheritability can, meaning it's like it can also be used incorrectly in terms of associations and all that kind of stuff that are really important, and we're going to delve into in just a bit. Okay, so first, looking at just examples of the correlations of different traits between monozygotic twins and dizygotic twins. So here, just in terms of height, you can see that siblings, it's about 50%, likelihood that they'll have similar height. And whereas if it is, like identical twins, then it's really close, so like 90%. Nothing is pretty much 100% because of the pieces of, because of the role of the environment and elements of gene expression. So looking at each of these, you can get a sense of, okay, there's always higher heritability, the more genetically similar, but then some of them are closer than others. So anxiety sort of disorder is at sort of 32, and this one is at 52. So these are basically like 20 points sort of, or the correlation is different. So it's like significantly less predictive, but then others have this relationship here is actually more significant, is actually of greater distance, in the sense that here temperament is basically 22% heritable, but then when you are getting up to but when you are monozygotic and identical twins, it's virtually like it's basically twice as likely that you'll have similar temperament. So so looking at these differences here, the further apart they are, then the greater the difference basically in aspects of genetic variation and environment. And they will also do these studies by looking at twins adopted and going to different locations. I mean, there's like twin studies are really quite, they're all over the place, and just in different ways to kind of track these different experiences of people that are genetically similar versus, siblings being raised in the same environment. All right, so then getting back to this behavioral genetic space of this, everything is within the behavioral genetics, looking at the role of the genes in behavior. Researchers are looking at complex components of behavior and mental processes. These are some of the things like personality, political affiliation, economic preferences, risk-taking or thrill-seeking. So they have a particular hypothesis, the reward deficiency hypothesis. This is this idea that there's a general deficiency in dopamine, so the subjective experience of rewarding behaviors is just not quite as subjectively great. So so again, like kind of thinking about two people who'd be going through the same sort of like thrill-seeking event, based on their genes, could experience the, like the level of thrill really quite differently. And so people who experience a lower thrill, or just not, again, as heightened dopamine, then might be trying to kind of seek out more sort of risk-taking or thrill-seeking to accommodate for that. And so the reduced subjective experiences rewards leads to higher incidences of boredom, and more likely to seek external stimuli from the environment to elevate dopamine. And so this is, so this can come out in behavior in various ways. And then there's also components of the genetics that are thought to be involved in religiosity and spirituality. And so there is research that is delving into this space. Some people really don't like the idea of like, wait, the fact that I'm religious is due to some aspects of my genes, but it's really also a lot of, epigenetic factors in terms of, like, even if you're living in a cultural group, aspects of cultural groups, then some of them have religious affiliations or morals or values. And the more likely, you are to, like in these spaces that can promote these things and promote the idea of support through religiosity and spirituality that can create sort of like support expectations, or, and I'm just postulating here, guys, just that, thinking about, okay, then how this person might feel more comfortable, sort of living in this sort of, in this space of sort of religiosity and structure and sort of, or spirituality. So our genes really underlie a lot of stuff. And people don't like to think that they would also impact aspects of political affiliation, but they do. And there are differences, in some core aspects of differences, and at least in how that are attributed to some differences in preference for one political affiliation or another. And so there's all sorts of elements that are looking into here, and then some of, so there's a range of implications of these things. All right, so that is the end of this section, and we'll be moving on to the last part, where we'll be getting into natural selection evolution, and then heading into some of the real implications of like how natural selection evolution has been used, and how beneficial it is, but also in misunderstandings and misattributions of it to also kind of perpetuate to fields like epigenetics, excuse me, not epigenetics, eugenics, that are an inappropriate use of natural selection. So coming back to this just in a few minutes.